सेकेंड चैप्टर है एनालिसिस ऑफ पैथोलॉजिकल कंस्टिट्यूएंट ऑफ यूरिन. Substances which are not present in easily detectable quantities in urine of healthy beings but are known to occur in urine under certain disease conditions are called abnormal or pathological constituent of urine. Before proceeding to detect the presence of abnormal constituent, note the physical characteristics, color, appearance, sediments, and specific gravity and chemical reaction with litmus paper. The abnormal constituents which are routinely looked for in urine are reducing sugar, acetone bodies, protein, blood pigments, bile pigments, and bile salts. Reducing sugar, acetone bodies, protein, blood pigments, bile pigments, and bile salts. Reducing sugars. Even normal urine contains a trace of glucose and glucuronides, but their amount is too small to cause a reduction in Benedict's test. A sample of urine which reduces Benedict's reagent under the conditions laid down for the test may be looked upon as containing reducing sugar unless otherwise proved. Even normal urine contains trace of glucose and glucuronides, but their amount is too small to cause reduction in Benedict's test. A sample of urine which reduces Benedict's reagent under the conditions laid down for the test may be looked upon as containing reducing sugar unless otherwise proved. A positive Benedict's test does not necessarily indicate the presence of glucose in urine, for such a result may quite possibly be due to the presence of other reducing sugars such as lactose in case of pregnant and nursing women, fructose in fructose-urea, galactose, fructose in fructose-urea, galactose in galactose-urea, and peptones, pentose in pentose-urea, homogenesic acid in alkapton-urea, glucuronides, and mucin. However, positive Benedict test is usually taken for glucose unless other conditions are suspected and proved. If mucin is suspected, Benedict's test should be repeated after removing mucin with choline. If the urine is from a pregnant or nursing lady, presence of lactose may be suspected. The identity of different reducing sugar, glucose, lactose, galactose, fructose may be established by performing Selbynoff test, Osazone tests, fermentation test, or chromatography. Benedict's reduction test to about 5 ml Benedict's reagents in a boiling tube, add 0.5 ml about 8 drops of the urine. He hold the test tube firmly with a test tube holder and boil the contents for 2 minutes over a steady small flame. A turbidity or precipitate of either green, yellow, or red color indicates the presence of reducing substance in the urine. Usually glucose. The various color of the precipitate depends upon the concentration of sugar in urine, and therefore a rough estimate of sugar in urine can be made in the following ways. Light green, turbidity 0.1 to 0.5%, green precipitate 0.5 to 1%, yellow precipitate 1 to 2%, red 1 to 2%, red precipitate above 2%.
Note: the color variation in relation to the concentration of sugar is seen in urine only and not in pure sugar solution in water. The difference is probably due to creatinine, another constituent of urine. Caution: Boil urine for at least 2 minutes. While boiling, the contents of the test tube have the tendency to sprout out. Therefore, keep the test tube shaking while holding it in inclined position at a place near the flame, which does not overboil it. Blue solution, green और येलो पीपीटी, ऑरेंज और रेड पीपीटी. इसके बाद लास्ट में कैसा मिलता है, ब्रिक रेड पीपीटी. ब्लू सॉल्यूशन में नन, कोई शुगर नहीं. ट्रेस ऑफ रिड्यूसिंग शुगर, ग्रीन और येलो पीपीटी. ऑरेंज और रेड पीपीटी मॉडरेट, और ब्रिक रेड मिल रहा है तो लार्ज अमाउंट ऑफ रिड्यूसिंग शुगर जो है प्रेजेंट है. A strip test for glucose in urine. Nowadays, a special paper strip impregnated with hexokinase or glucokinase enzyme and chromogen are available from many commercial companies to test the presence of glucose in urine. When the strip is dipped in urine, some of it is sucked up the strip where glucose is oxidized by enzyme to produce hydrogen peroxide. Hydrogen peroxide when reacts with the chromogen to give green coloration at the tip of the strip. On the basis of intensity of color produced, concentration of glucose in urine can be judged by comparing the color with a standard chart provided by manufacturer. The test is positive only when glucose is present, but not when any other reducing sugars are present. Similarly, paper strip test is available for a number of other abnormal constituents of urine such as albumin, bile salt, bile pigment, urobilinogen, blood pigment, acetone and acetoacetic acid. यूरिन टेस्ट स्ट्रिप इधर है लिस्ट, क्या क्या इस स्ट्रिप से टेस्ट हो सकता है. ग्लूकोज, बिलिरुबिन, कीटोन, यूरोबिलिनोजेन, प्रोटीन, पीएच, ऑकल्ट ब्लड, स्पेसिफिक ग्रेविटी, नाइट्रेट, WBC. ये नेगेटिव, ये पॉजिटिव. द मल्टीस्टिक्स यूरिन टेस्ट स्ट्रिप शोइंग द मैन्युफैक्चरर कलर्ड स्केल. यहाँ पे क्या है, LEU ल्यूकोसाइट्स 2 मिनट के लिए करते हैं. NIT नाइट्राइट के 60 सेकेंड. टाइमिंग कितना देर रखना है. सो URO यूरोबिलिनोजेन 60 सेकेंड. PRO प्रोटीन 60 सेकेंड. PH 60 सेकेंड. BLO ब्लड 60 सेकेंड. SG स्पेसिफिक ग्रेविटी 45 सेकेंड्स. KET कीटोन 40 सेकेंड. BIL बिलिरुबिन 30 सेकेंड. GHU ग्लूकोज 30 सेकेंड. एक्सपेंडेड व्यू ऑफ यूरिन स्ट्रिप कंपैरिजन चार्ट. अब आते हैं एसीटोन बॉडीज पे. When glucose metabolism is slow as in diabetes mellitus, or when carbohydrate is not available in the starvation, fat is excessively catabolized. As a result of it, acetoacetic acid and its derivative beta hydroxybutyric acid and acetone are excreted in urine. This condition is called ketosis, and these three compounds are together called acetone bodies or ketone bodies. The excretion of ketone bodies in urine is called ketonuria. Experiment number 10. Rothraj test for acetone bodies. Saturate 5 ml urine with respect to ammonium sulfate by shaking it with excess of solid ammonium sulfate until some remains undissolved. Add 2 drops of freshly prepared 5% solution of sodium nitroprusside or a small crystal of sodium nitroprusside and shake to dissolve in the urine and mix. Then gently layer 2 ml ammonium hydroxide over the urine. At the junction of urine and ammonia layers a permanganate color ring appears. Okay, add 2 drops of freshly prepared 5% solution of sodium nitroprusside or a small crystal of sodium nitroprusside and shake to dissolve in the urine and mix. Then gently layer 2 ml ammonium hydroxide over the urine. At the junction of urine and ammonia layer a permanganate color ring appears. If acetone bodies are present on mixing the two layers, the whole solution assumes this color. The test cannot be regarded as negative until the mixture has stood for 10 minutes. Without giving this color, the permanganate color reaction is due presumably to the formation of ferromagnetic cyanide with the isonitro compound of the ketone, or the formation of such an ion with the isonitrosamine derivatives of the ketone. Formation of such an ion with the isonitrosamine derivatives of the ketone. गरहर टेस्ट फॉर एसिटोएसिटिक एसिड. अभी हम लोग कीटोन बॉडीज के लिए किए, एसीटोन बॉडीज के लिए किए, और एक है एसिटोएसिटिक एसिड के लिए स्पेशली गरहर टेस्ट फॉर एसिटोएसिटिक टेस्ट. तो इसमें ये तो देखते हैं क्या है इसमें. इफ रोथराज टेस्ट इज पॉजिटिव, प्रेजेंस ऑफ एसिटोएसिटिक एसिड कैन बी टेस्टेड बाय फेरिक क्लोराइड रिएक्शन. To about 5 ml urine in a test tube, add 5% ferric chloride solution. 5 ml urine में 5% ferric chloride solution drop by drop add करना है till no more precipitation of ferric phosphate is formed. filter करना है. To the filtrate add some more ferric chloride. A brownish red color appears if acetoacetic acid is present. Note, a similar color is given by large number of substances such as aspirin, antipyrine, salicylates, etc. If the urine is boiled, acetoacetic acid is converted into acetone, but the other substances are not destroyed. A urine which gives a positive test before boiling and negative after boiling indicates the presence of acetoacetic acid. Ferric chloride test is also done for detection of phenylpyruvic acid in urine. In this case, a green or blue color appears for a few minutes or two. Phenylketone urea is due to serious inborn error of phenylalanine metabolism. अब आते हैं प्रोटीन में. Presence of heat coagulable protein in urine is called proteinuria, or more loosely called albuminuria. In such urine, generally both albumin and globulin are present. Sometimes Benz-Jones protein and immunoglobulin or protease may be present. Benz-Jones protein appears in urine in case of multiple myeloma. It has the characteristic property of coagulation between 40 to 60 degrees Celsius, of redissolving on further heating and reappearing on cooling to below 60 degrees Celsius. Of the various tests for protein in urine, the boiling test is most commonly employed. The sulfosalicylic acid test is strongly recommended for routine work. A positive test should be checked by the boiling test. Of the various tests for protein in urine, the boiling test is most commonly employed. The sulfosalicylic acid test is strongly recommended for routine work. A positive test should be checked by the boiling test. Sulfosalicylic acid test, experiment number 12. To 2 ml of clear urine, filter if not clear. 2 ml क्लियर यूरिन लेना है, अगर क्लियर नहीं है तो फिल्टर कर लेना है. एड या फ्यू ड्रॉप्स ऑफ सल्फोसैलिसिलिक एसिड, कुछ बूंदें या फ्यू ड्रॉप सल्फोसैलिसिलिक एसिड का ऐड करना है. एक टर्बिडिटी या टर्बिडिटी इंडिकेट्स द प्रेजेंस ऑफ प्रोटीन. दिस टेस्ट इज पॉजिटिव फॉर एनी प्रोटीन, हेंस presence of coagulable protein in urine must be confirmed by boiling it. Boiling experiment number 13, boiling test for coagulable protein. Take 5 ml urine in a test tube and adjust its reaction to pH 5.4 by adding 2% acetic acid or 2% sodium carbonate and using chlorophenol red indicator. Divide it into 2 portions. Heat 1 portion to the point of boiling and no more, and look for the turbidity. Comparing it with the unheated portion, turbidity may be due to coagulable protein, albumin and globulin or earthly phosphate. Add a drop or 2 of 33 acetic acid to the hot turbid urine. If the turbidity disappears, it is earthly phosphate. If not, it is protein. Modified boiling test. Fill a test tube 3/4 full of clear urine. If the urine is turbid, filter it. Before performing the test, heat the upper 1/3 of the test tube over a small flame. The turbidity is seen in the heated portion of the urine, well contrasted with the unheated lower portion. If coagulable protein or phosphate is present, the presence of protein or phosphate can be distinguished by adding a drop of 33% acetic acid to the urine, in which earthly phosphate dissolves but not the protein. Even if turbidity does not appear on heating, add 1 to 2 drops of acetic acid to hot urine. Sometimes turbidity appears now when protein is present. Take 5 to 10 ml urine and boil the upper portion. Check for cloudiness and add 2 to 3 drops of glacial acetic acid. Reboil the upper portion. Cloudiness means presence of protein. अगर क्लियर हो गया तो absence of protein. Modified boiling test. अब आते हैं बाइल में. Constituent of bile such as bile salts and bile pigments appears in urine of persons suffering from obstructive jaundice or hepatic jaundice. Urine containing bile pigments appear yellow or greenish yellow in color. Experiment number 14, Hayes test for bile salts. Sprinkle a little flour of sulfur over the urine contained in a test tube and note if sulfur particles sink, which indicates the presence of bile salts. Control for comparison or positive test. Hayes test for bile salts. अभी सल्फर पार्टिकल डूब गया, सिंक, इसका मतलब उसमें बाइल साल्ट है, नहीं डूबा तो बाइल साल्ट्स नहीं है. एक्सपेरिमेंट नंबर 15, Faucet's test for bile pigments. Take 15 to 20 ml of urine in a boiling tube and add to it 1 to 2 drops of saturated magnesium sulfate solution and about 5 ml of 10% BaCl2 barium chloride. Mix the content and let the tube stand for a while for the precipitate to settle down. Decant the supernatant and filter the rest. After the filtration is complete, spread the filter paper over a tile and add 1 drop of Faucet's reagent. To 1 part of precipitate. On the filter paper, bile pigment is indicated if blue or greenish blue color appears at the place where Faucet's reagent has been added. Experiment number 16, Tests for urobilinogen. Urobilinogen is formed from bile pigments in intestine and is absorbed to be excreted in urine. Urobilinogen is excreted in small amounts in normal urine, but is excreted in greater quantity when large amount of bile pigments reach intestine, as in hemolytic jaundice. Absence of urobilinogen from urine indicates obstructive biliary passage, obstructive jaundice. To 5 ml urine, add about 0.5 ml Ehrlich aldehyde reagent. 5 ml urine and about 0.5 ml Ehrlich aldehyde reagent, 2 gram of para-dimethyl aminobenzaldehyde dissolved in 48 ml conc HCl and volume made to 100 ml with distilled water. 2 gram of para-dimethyl aminobenzaldehyde dissolved in 48 ml conc HCl and made to 100 ml with distilled water. Mix and allow it to stand for 2 to 4 minutes. Normal urine give
gives out a pink color, but a distinctly red color suggests the presence of increased amount of urobilinogen. This test should be performed on fresh urine only. Next is blood pigments. In certain pathological conditions, blood appears in urine. The presence of blood can be recognized by reddish coloration of urine and by microscopic examination for red blood cells if it has not been hemolyzed. Chemically, hemoglobin or its derivatives can be demonstrated by benzidine test. Experiment 17, benzidine test. Boil some urine first and allow it to cool. In a dry test tube, dissolve a nif point of benzidine in about 1 ml glacial acetic acid and then add to it 1 ml hydrogen peroxide. Add 5 to 10 drops of urine in the test tube. The benzidine solution will turn deep blue if blood pigment is present. In the urine, if the benzidine solution turns blue before adding the urine, prepare another solution of benzidine. The test is very sensitive, and any test tube which once obtained blood and has not been cleaned thoroughly will give positive test. It will be noted that the blue color of benzidine produced by blood pigment is temporary and changes to brown within a few minutes. Benzidine test. Now questions and answers.